Today, after over a year, we finally wrap up the series on the hallmarks of aging. And in my opinion, this hallmark may very well be the hallmark that has the biggest impact on aging, yet has the most potential to be improved. Stem cell exhaustion, the depletion of our pool of stem cells. Before I begin, I'd just like to remind you guys that I'm in the process of creating an online course on longevity. And if you'd like to be on the mailing list to receive updates on the course, check out the card up here. I started this series on the 9 hallmarks of aging over a year ago with my first video on mitochondrial dysfunction. Today, I finished the series by talking about stem cell exhaustion. I'm also doing this video at this time because in a few weeks, I'm going to be posting a video about a peptide called GDF11 and its amazing longevity attributes. And you'll need this video as background. Now, in my opinion, this hallmark may have more direct impact on aging than any other hallmark. Think about it. All the other hallmarks cause some form of damage to the cells. But as long as you can simply replace that damaged cell with a brand spanking new cell, you don't age. Damage happens all the time, but it's once you lose the ability to replace old and worn out cells with new ones that you begin to age, which is what makes stem cell exhaustion so important. Stem cells are the cells that replace old cells that are malfunctioning. And once you exhaust your supply of stem cells, that replacement process grinds to a halt. But if stem cell exhaustion is so important, why is it one of the last hallmarks? That's because the hallmarks aren't listed in order of importance, but rather starts with the primary initial damage and proceeds through a chain of events that eventually leads to aging. And stem cell exhaustion is at the end of that chain. And that's good news. The more things that have an impact on a hallmark, the more opportunity there is to make improvements. Almost all the other hallmarks have an impact on stem cell exhaustion. To really understand what a stem cell is, we need to start at the beginning, when life is first created. When an ovum is fertilized by a sperm cell, a zygote is created. This zygote contains all 26 pairs of chromosomes, half donated by the father and half coming from the mother. This single-celled zygote starts dividing and replicating itself, and it becomes a blastocyte, a ball of cells that contain the first stem cells. Now, these are called embryonic stem cells, and each stem cell has the potential to become any type of tissue or cell in the body. This ability or potential is called pluripotency. When a stem cell transitions from this potential to actually becoming a specific type of cell, that's called differentiating. As the fetus grows, the vast majority of these stem cells differentiate into a specific type of cell, thereby losing the ability to differentiate. But a few retain this potential, the capacity to differentiate and become what's known as progenitor cells or multipotent stem cells. A cell's potency is its ability to differentiate into other cell types. The more different types of cells that it has the potential to become, the higher its potency. A multipotent stem cell is less potent than a pluripotent stem cell. Embryonic stem cells differentiate into progenitor cells, which can only differentiate into a limited class of cells. For example, hematopoietic stem cells are a type of progenitor cell that can only differentiate into the various blood cells. Throughout our lives, we retain a pool of these progenitor cells, these multipotent stem cells, whose job it is to constantly replace most of the somatic cells in our bodies as they become aged and worn out. This is what keeps us young. Somatic cells are all the cells in our body except for the germ or sex cells and the stem cells. Most somatic cells enter a phase called G0, where they are still metabolically active, but they don't replicate or proliferate. Some of them never proliferate, but most of them still retain the ability to proliferate if needed, as in the case of injury or damage. Once these somatic cells reach the end of the cell life cycle, they die and are recycled, and they're replaced by progenitor stem cells. For example, hematopoietic stem cells in the bone marrow constantly replace our supply of red and white blood cells and platelets. Muscle stem cells, called satellite cells, repair and regenerate new muscle tissue. Stem cells in the base of the epidermis replace dead skin cells. The nervous system and the heart tissue can be partially renewed, but they undergo minimal renewal in comparison to the rest of the body. Throughout life, these stem cells retain not only the ability to differentiate and become replacement cells, they can also proliferate, producing more stem cells as others are used up. This is why throughout our younger years, our supply of stem cells remains high, and we're able to recover from injuries or illness. We can recover from workouts more quickly and more effectively. But as we age, this pool of stem cells becomes depleted, and those that remain lose their capacity for renewal. Now, this is stem cell exhaustion, and immunosenescence, sarcopenia, and osteoporosis are just 3 examples of conditions that arise from stem cell exhaustion. Also, that thin, wrinkled, crepey skin that we all develop as we age, stem cell exhaustion. All right, let's take a look at some of the things that can drive stem cell exhaustion, that can take a system that's functioning just fine and turn it into a system that's failing. There's a surprising number of different causes that can trigger stem cell exhaustion, and most of them are occurring simultaneously. Rather than just a single driver, it's all of them, or at least most of them, meaning that just addressing one probably won't cut it. One of the primary reasons in stem cell exhaustion is damage to the DNA. This is when the DNA of a single stem cell becomes damaged, and if that happens and the damage doesn't get repaired and there becomes enough of it, that stem cell will probably die or become senescent, instead of proliferating and differentiating like it's supposed to. And the stem cell pool just got smaller. Now, I talked about a lot of the ways that damage can happen to DNA in the video on genomic instability, so I'm not going to go over that here. But there's a few different ways that DNA damage can build up in a stem cell. First, damage can accumulate over time, and not in just one cell. If the damage isn't repaired, it can be passed on to daughter cells. Second, the rate at which damage occurs increases. Damage to the DNA usually occurs due to stresses, either internal stresses or external stresses. If those stresses go up, so does the rate of damage. The third way that damage can build is if the rate of repair in response to DNA damage goes into decline, if the rate of repair goes down. As we age, DNA damage repair, or DDR, falls into decline. With age, repairs happen less often and are less effective. But there's another factor that comes into play. Throughout most of our lives, most of our stem cells are dormant, and they're in a state of quiescence. They only become activated when the stem cell enters the cell cycle, when the cell replicates or proliferates. As long as the stem cell is in a state of quiescence, the repair processes don't get activated. They usually only get triggered after the stem cell is activated. Now, stress happens all the time, and the stem cell can become damaged during the quiescent period. This damage continues to build up until the stem cell enters the cell cycle and repair processes get activated. Now, when we're younger, this is okay. Our repair processes are so effective that the DNA damage to the stem cell can be repaired perfectly. And because we're still relatively young, not that much damage is built up. But when we're older, it's a problem. As our repair processes go into decline and the damage continues to build, at some point, the damage overwhelms the repair processes and crosses a threshold. And instead of getting repaired, the stem cell either dies or becomes senescent. Here's a paper that looked into that very process in hematopoietic stem cells, or HSCs. Independent of age, quiescent HSCs showed reduced expression of DNA repair genes to accumulate DNA damage that is repaired only when the cells enter the cell cycle. As many HSCs remain quiescent for much of adult life, the continuing accumulation of damage in these cells may eventually exceed a threshold that impairs stem cell function in aged animals. But damage to the DNA is not the only way that stem cells can age and start malfunctioning. Loss of proteostasis, another hallmark, can also drive stem cell exhaustion. Regarding stem cell function, Proteostasis has been implicated as an important determinant of stem cell maintenance through studies in HSCs showing the deletion of autophagy-related gene 7 increases ROS levels, or reactive oxygen species, and depletes HSCs. Autophagy is part of the proteostasis degradation machinery and is controlled in part by the mTOR and the AMPK pathways. Epigenetic remodeling can also drive stem cell exhaustion. So can telomere attrition. Repeated demands on stem cells to generate replacement cells over an entire lifetime may cause an accumulation of cell-autonomous defects, including telomere erosion, mutations in epigenetic dysregulation, and eventual replicative exhaustion. Mitochondrial dysfunction has also been suggested as a cause of stem cell exhaustion. So has oxidation caused by ROS and inflammation. The point is, all of these factors contribute to the loss of function in stem cells. But loss of function, for whatever reason, doesn't always lead to death of the stem cell. Sometimes stem cells become senescent. Senescent stem cells leave the cell cycle, but they don't die. They don't proliferate and they don't differentiate, but they're still alive. And they can secrete inflammatory compounds called senescent-associated secretory phenotypes, or SASPs, that can further cause inflammation and can induce other stem cells to become senescent. All these different factors can interact with each other in a vicious circle that builds and builds the older that we get. Which brings us to the question, what impact does stem cell exhaustion have on the aging process? And it should be pretty obvious. Without stable populations of proliferating stem cells, tissues and organs lose their ability to recover from damage and begin to fail. If the human body loses the ability to replace damaged cells as they die, then the tissues begin to malfunction, to break down and deteriorate, and eventually to fail. Since stem cell exhaustion is in the final category of hallmarks, I guess you could say that while there are a lot of upstream things that can go wrong and impact stem cell exhaustion, in the final analysis, it's this hallmark that may ultimately be responsible for how well we age. So, what can we do about it? What can we do to boost our stem cell pool? How can we avoid stem cell exhaustion or restore stem cell pool if it does become depleted? Well, because this hallmark is at the end of a chain of events that leads to aging, breaking any part of the chain that's upstream will improve the stem cell pool. Fixing upstream hallmarks will automatically improve downstream hallmarks. For example, simply changing your lifestyle can have a big impact on this hallmark. Eating better and being more active can improve epigenetic methylation patterns and boost mitochondrial function, both of which can impact stem cell exhaustion. So can supplementing with NAD precursors. Increasing NAD+ supplies will improve mitochondrial health as well as activating sirtuins for DNA repair. It can also help prevent stem cell senescence. Rapamycin, for those who can get it, has been shown to restore self-renewal and hematopoietic potential of aged stem cells by inhibiting the mTOR pathway and activating autophagy. Taking spermidine can also trigger autophagy. Clear out senescent cells by using senolytics. Remove senescent cells and remove one of the major sources of inflammation, SASPs, and a major cause of stem cell exhaustion. But what if, rather than clearing out senescent stem cells, there was a way to reactivate them, to turn them back into viable stem cells? Well, there's a compound that I'm becoming more interested in that looks like it might do just that. Growth differentiation factor 11, or GDF11, appears to reactivate senescent stem cells. Now, this could have a big impact on the stem cell pool, and taking GDF11 appears to have a number of beneficial anti-aging effects, most of them associated with improving stem cell exhaustion. GDF11 is so interesting that I'll be doing a video on this peptide in a few weeks. Finally, in the future, another option might be to boost the stem cell pool by simply replacing the stem cells. In 2016, Shinya Yamanaka discovered 4 factors that could induce pluripotency, the same potency that is in embryonic stem cells, in somatic cells, by resetting the epigenetic environment to a more youthful state. Now, this is still experimental, and it's not entirely successful, but the idea is that the patient's own somatic cells would be harvested. These Yamanaka factors would be applied in vitro, and the cells would become induced pluripotent stem cells. These stem cells would then be infused back into the patient, and since they contain the patient's own DNA, these stem cells would become part of the stem cell pool and replenish it. While I'm not trying to belittle the importance of the rest of the hallmarks of aging, in my opinion, restoring the stem cell pool is one of the most promising things that you can do to defeat the aging process. And there seems to be a lot of different paths that can lead to that. My goal is to pursue as many of these paths as possible and hopefully restore my stem cell pool to a more youthful level. I hope that you've enjoyed this series on the hallmarks of aging and this video on stem cell exhaustion. Now, if you haven't watched the whole series, then you should check out this playlist. That's it for me. I'm out of here. Catch you next time.